Hello students, today I am here with the presentation where we'll discuss about the viruses, their infections and their treatments. Nowadays this topic is very relevant in today's scenario as we all are suffering with the consequences of COVID-19 infection worldwide. The antiviral drugs are serving three main purposes, or you can say we have the three main approaches for the viral infections. First is the treatment of viral infection through vaccination. Second is the antiviral chemotherapy. Here we'll discuss mainly about the antiviral chemotherapy. And third approach is the stimulation of host resistance mechanism. Means if the host resistance mechanism is stronger, that will help us or help the patient to fight well against the viral infections. Viruses, you know all that they are like a parasites and they live inside the body and uses the biochemical mechanism of the host. And the products they utilize biochemical products of the host, and by that they maintain their life. To reproduce, the virus must enter the host cell. Now if virus need to multiply, virus needs a host body. Take over the host cell's mechanism for nucleic acid and protein synthesis. So it utilizes the host cell's mechanism, means if it is in human body, it utilizes the human body's mechanisms, right? And then utilizes the mechanism and use it for making their nucleic acid and proteins, and direct the host cell to make new viral particles, and like that new viral particles developed. Drugs can target virus specific steps. I'll discuss in the coming slides that how this virus is actually infecting. And if you follow from beginning, the every process is a very potential target for the drug treatment. So you can target the cell penetration stage, uncoating stage, reverse transcription stage, virus assembly stage, or the maturation stage. And all of these stages we can block and inhibit the viral processes. So like these drugs will serve for us. We'll see the classification of viruses. Here, this is not a classification of drugs. I'm talking about the classification of viruses. These viruses have been classified usually by two types. One we call it as an DNA viruses, another is called as the RNA viruses. So, if you see a common structure of virus, you can see their nucleic acid, which is called as the core of the virus particle, and that nucleic acid, this can be RNA, can be DNA. This nucleic acid is enclosed by, covered by one kind of protein covering, which we call it as a capsid. So this is a normal structure what the virus looks like. And these viruses are of two type. One DNA viruses and there is RNA viruses. So on the based on their morphology and cellular site of viral multiplication or other characteristics, these divisions have been made. So DNA viruses, if you see the examples of it, the DNA viruses includes adenoviruses, usually cold virus and conjunctivitis infections caused by the adenoviruses. Hepatadenoviruses, that is causing the hepatitis B. Herpes viruses, you know that cytomegaloviruses, chickenpox, shingles, these all been caused by the herpes viruses. Herpes virus is also called as herpes simplex viruses, HSV. Papilloma viruses, they are usually causing the warts. And pox viruses, they are usually causing the smallpox. So these all are the DNA virus category. Next is the RNA viruses. The RNA viruses includes arboviruses. You know, examples are tick-borne encephalitis, yellow fever. Arenoviruses, for example, Lassa fever and meningitis. You might have heard about the Lassa fever. Orthomyxoviruses, for example, influenza. Paramyxoviruses, measles and mumps. Picronaviruses, polio, meningitis and cold. Rhabdoviruses, rabies. Rubella viruses, German measles. Retroviruses, AIDS. So, like these viruses are there which are causing various infections. We have to understand the viral replication process as well. Usually, the viral replication process is classified into 5 different processes. As I told you in beginning, that every phase of the virus particle process is a very potential target for the treatment. So here there are 5 phases, means 5 potential targets. So these phases, first is the attachment and penetration. The virus which is coming to the host body need to identify the host cell. After the identification, the viral particle goes and bind with it, which we call it as an attachment. After the attachment, it penetrates inside the cell, right? So that is the attachment and in penetration. After entering inside, it will go for uncoating. The main functional material is the genetic material, what called the nucleic acid of the virus. So why nucleic acid has to come out, so the outside virus coat will dissolve. So uncoating will happen, then synthesis of viral component, then viral need to make more components, their nucleic acid, more division of their nucleic acid, multiple copies, assembly of virus particles. Once they'll make their new nucleic acid, and as well as they will make the required components of proteins, they will try to assemble themselves. So assembly of virus particle will happen, and then after that, once the particle is been formed, the new particles have been synthesized, now they'll release the virus. So like that, 5 different phases, and every phase is a one potential target, right? You can have attachment inhibitors, penetration inhibitors, uncoating inhibitors, synthesis of viral component inhibitors. Like that, you can make different drugs to inhibit all these processes, and we have most of the drugs. This is the one diagram of the life cycle of a virus. This is shown as an reverse transcriptase virus, or you can say the virus which is responsible for the HIV. So here we'll see the structure. This is also a common structure of the antivirus. You can find that here you can see inside the reverse transcriptase enzyme is there, which is playing the role to make this RNA later into the DNA. So here this is RNA inside. The outer covering of protein is called capsid. There is one protease enzyme. This protease enzyme helps the virus to penetrate into the cells. Here is an integrase which help the virus particle to integrate and make a mature particle. And this is very important two things you can find here, GP120 and GP41. So this is these two very important proteins which are called as the envelope proteins, and these proteins are helping the virus particle to attach to the host. So usual process is after that, once the particle goes inside the cell by the help of the clathrin protein. Clathrin proteins are making the vesicle for the virus, a safe zone by which it goes inside safely, and will avoid any attack by the cell. So it reaches safely inside the cell. Once it enters into the cell, this is clathrin protein. Once it enters into the cell, it go first for the removal of the protein envelope. The first envelope has to be gone, remove. After that, the reverse transcription process will happen. The viral particle will RNA will convert into viral DNA. Then they will enter into the cell's nucleus, and there it will integrate itself. Means this viral particle will integrate into the host DNA. See the light greenish color is introduced into the host DNA. Right now the host DNA will get multiply, means your own machinery started working for virus. Virus has hijacked your machinery. So now here after this you can see the viral proteins have been formed, and after this the complete things will get into the assembly, and once the assembly is done the virus has been released outside of the cell. So like that these commonly the viruses are playing the role. This is another diagram for the common viruses. You can see it's an RNA viruses here. Attachment, you can see it goes through endocytosis inside, then the coat has been dissolved, then RNA comes out. Once the RNA comes out, RNA directly can go for the protein synthesis by using the endoplasmic reticulum and the ribosome, and by that it will produce the lot many viral particle and released outside. Here one more diagram which is also showing you the similar thing. You can see the first stage is an attachment, second is the membrane fusion, means that it fuses with the membrane. Second, release of viral DNA through nuclear pores, it's enter into the nucleus. Then transcription of the viral mRNA, then mRNA will be formed. Then synthesis of viral protein by host cells ribosome, that is in the fifth place. Sixth place, replication of viral DNA by viral polymerase. It has been shown next to the 5. I think here you can see. And the 7, assembly of virus particle. Then the 7th way the particle will assemble, and 8th is the budding and release of the progeny virus. Like that these viruses will get formed. Replication, here has been shown one specific example of the life cycle of the influenza virus, right? See, it's a process is almost similar. You can go through with it. Now, just for your to give you a little more insight, I have added about the SARS-CoV, which we call it as COVID-19. So this is a SARS COVID-19 infection. Let's see what is unique about this. In this process, this is little bit different process here. You can find out the virus particle, which looks very different than the other virus particles. It is having a single RNA molecule inside, and that single RNA molecule plays a role. It requires certain proteins to form, and these proteins are having very specific role into it. Once it enters into the host cell, it releases its RNA particle, and then this RNA particle will forms the RNA polymerase enzyme. This RNA polymerase enzyme will form the one another RNA strand, which is called as a negative strand of it. Now this negative and positive strand both will form the more positive strands, and now these more positive strands will make different proteins, for example N protein and M proteins, and then other proteins like S protein and HE proteins. So now these all proteins will enter into the nucleus of the cell, and there they integrate and make a one virus particle, and this way by using the Golgi apparatus it will make more copies of it, and then the Golgi, and then it will release outside of the cell. So like that the COVID-19 virus also plays a part. You can read it the sideline details of it. In the next slide, this is also a little more descriptive life cycle of the SARS-CoV. Here you can find a little detail about the ACE2 receptors. Usually the coronavirus binds on the ACE2 receptors, and these ACE2 receptors, once it binds in it, sorry, once it binds in, it allows the infusion or the fusion of the virus into the host membrane, cell membrane, and then it enters through endocytosis. There it forms the endosome, and then it reaches there. After that there will be uncoating. Uncoating means the outside coat will be dissolved, and the inside what called the genetic material comes out. After that, it will go into the translation process. You know very well what the translation process is. Then after the translation process, when it uses its RNA and go for the protein synthesis, that is a process of transcription. Sometime it takes the process of transcription, then again translation, and then it makes the various viral proteins. Sometime by using the directly these proteins, it will go for the assembly, right? And once all the required means genetic material like nucleic acids have been formed, as well as some required proteins have been formed, now this will go for assembly, and after assembly it will release from the body as a matured particle. So this is the way life cycle of SARS-CoV is also there. Now we'll see the drugs which are playing the role in it. The virus particle, what I have explained you initially, has the various proteins in the structure. As you see that the transmembrane glycoprotein is there at the end, at the top of it, that called the GP41. An extracellular glycoprotein was there at called it at GP120, right? And the major structural proteins are there, which we call it as P24 capsid proteins, like. And then lipid layer is there, one protease enzyme is there, one reverse transcriptase enzyme is there, RNA is there, integrase enzyme is there, and nucleocapsid is there, which is made up of the protein 17 called P17. So like that, these different proteins are there. Why I'm telling you these proteins? Because there are the drugs which are inhibiting these particular proteins, and affecting the virus maturation and the virus assembly, or the various phases of the virus development. Now we'll see the drugs here. First is the anti-herpes virus. Idoxuridine acyclovir, valacyclovir, femicyclovir, gencyclovir, and foscarnet. So these are the anti-herpes simplex virus, which is call it as an HSV virus. Second is the antiretroviruses. In this, we have two categories: nucleoside reverse transcriptase inhibitor called NRTIs, and non-nucleoside reverse transcriptase, which we call NNRTIs. So NRTIs are having very potential drugs: zidovudine, diadenosine, gelcitabine, stebudine, lamivudine, abacavir. And NNRTIs are having nevirapine, efavirenz, dalviridine, right? Third, next category is the protease inhibitors. These are ritonavir, indinavir, nelfinavir, squinavir, emprinavir, lopinavir, and many more. These are later I'll tell you that these some of these drugs are very much potentially used nowadays against the coronavirus infection. Anti-influenza virus, third category, where we have amantadine and remantadine. Non-selective antiviral drugs are also there. For example, ribavirin, lamivudine, adifovir, dipivoxel, interferon alpha. Here, this is a very important slide which tells you the virus life cycle, and will tell you where these drugs are acting in which particular phase. If you see the viral attachment and the entry time, there are these process has been blocked. I have told you that all these phases are a potential target. So first phase is the targeted by efuviridine, meroviroc, docozenol, and pelvizumab. Right, these drugs are inhibiting the viral entry. Some of these drugs are used for the coronavirus infections also. Then penetration process, which has been blocked by interferon alpha. Then we have the uncoating process, which has been blocked by the amantadine and remantadine anti-influenza drugs. Then we have the nucleic acid synthesis, a very very important part phase of the viral life cycle. So it is been blocked by NRTIs. We have also NNRTIs, acyclovir, phoscarnate. They all are playing the role for it. Next is the late protein synthesis. Means protein synthesis is happening for making the more proteins which are required for the viral survival. That is blocked by the protease inhibitors, right? Generally, HIV drugs are also comes in this category. Blocked by next category is the packing and assembly. After the packing and assembly, the virus has to release from the cell, and then once it comes out of the one cell, it starts spreading to the various cells. So it has been blocked by the neuraminidase inhibitors, influenza viruses. So generally, if you remember this, you'll remember the somewhat the link of mechanisms of these drugs. So we'll start the description of one particular drug of the one category here. So first one is the anti-herpes virus drugs. To treat herpes simplex viruses, which we call it as HSV, and varicella zoster virus, which is called VJV infection. So these drugs are used against the HSV and VZV viruses. Let's see about the acyclovir. So acyclovir is a guanine nucleoside analog, right? So you remember guanine nucleoside and the role of the guanine nucleoside in the DNA synthesis. It acts as a one of the base, right? So if you have a drug which looks like similar to the guanine nucleoside, it means this drug can bind in place of the guanine actually, and then it will disturb the DNA synthesis. So that is actually the mechanism of it. So acyclovir is a guanine nucleoside analog, most effective against HSV1 and HSV2, but it has some activity against VCV, CMV and EBV as well. Three are licensed. One is the against the HSV and VZV infections: acyclovir, valacyclovir and famiciclovir. These drugs have a similar mechanism of actions and very well tolerated these drugs. Acyclovir has been the most extensively studied. So we will see more detail about the acyclovir. So I'll tell you the acyclovir's mechanism of action here. Very nicely you can understand this mechanism. Acyclovir is converted to its active metabolite via 3 phosphorylation steps. Means to make the acyclovir active, it has to convert into its active metabolite, and that conversion happens by 3 steps. Number 1, viral thymidine kinase converts acyclovir to acyclovir monophosphate. Who is converting? Viral thymidine kinase. Means that is thymidine kinase which is present in virus, not in the host, right? So the virus thymidine kinase converts this acyclovir to acyclovir monophosphate. Then what happens? Host cell enzyme converts the monophosphate to the diphosphate. Now you see, both are playing the role here. First conversion done by the viral enzyme, second conversion done by the host enzyme. So host cell enzyme converts the monophosphate to the diphosphate and then to the active compound. So first conversion done by viral enzyme, and second and third conversion done by the host enzyme, and then it forms the acyclovir triphosphate, right? So now the acyclovir triphosphate has been formed. Because viral thymidine kinase has a much greater affinity for acyclovir triphosphate than does mammalian thymidine kinase. So don't think that thymidine kinase is not available for host. It is available for host. But the viral thymidine kinase has more affinity towards the acyclovir. That's why it not affects the means the host thymidine kinase is not acting for that, right? Only it means only virus, if it is a virus is there in the body, then only it will convert effectively into the its active metabolite. Otherwise it will be safe for the human body. Acyclovir triphosphate accumulates only in virus infected cells. Now you understood here that acyclovir converted into the acyclovir monophosphate by the herpes virus specific thymidine kinase. and then by the cellular kinases it is converted into the acyclovir triphosphate. Now this acyclovir triphosphate has a very special structure. It doesn't have the 3 dash hydroxy group. So this is an identification point. The acyclovir triphosphate doesn't have the 3 dash hydroxy group. Now the acyclovir triphosphate, which has been synthesized, acts as a competitive inhibitor for the incorporation of deoxyguanosine triphosphate DGTP into viral DNA. Means now acyclovir triphosphate has been formed, which is looks like similar to the guanosine base. So what will happen in place of the deoxyguanosine triphosphate, acyclovir triphosphate will bind into the viral DNA, right? It has been exchanged. Incorporated acyclovir triphosphate lacks 3 hydroxy group requires for the chain elongation. Now you know that if there is a no 3 group, you know always the growth is happening at the 3 to 5 end. So now there is no 3 when there is a no 3 it means what is going to happen? No chain elongation, right? Inhibits herpes virus DNA polymerase competitively, right? Then is a competition between that, and it inhibits that viral particle. Viral DNA polymerase becomes irreversible, bound to an acyclovir terminated DNA chain, unavailable for their activity for replication. Understood? So viral DNA polymerase become irreversible now, right? Because why? Because there is a competition now. You have got the new acyclovir triphosphate, which inhibits herpes virus DNA polymerase. Now herpes virus DNA polymerase cannot identify and work on it because 3 end is not there. So then the viral DNA polymerase become irreversibly bound to the N-acyclovir terminate and then terminate the DNA chain. And then because it is irreversibly bind there, so it is not further available also. Okay. Here, this diagram will also tell you the almost same thing, that how the acyclovir converts into monophosphate, diphosphate, triphosphate, and acts for the inhibits the viral DNA synthesis. Here is the structures, chemical structures of these drugs. The effect of acyclovir on host cell DNA synthesis is much smaller than its effect on the viral enzyme. That's what it's very safe. Concentration of acyclovir significantly beyond the therapeutic range are required to inhibit host cell growth. Means if you want that the acyclovir should affect the host or want to check the toxicity, then you have to give very high doses, more than the therapeutic doses, and then maybe it can affect on the host thymidine kinase, right? Now mechanism of resistance, this is also very important here. Herpes simplex and varicella zoster virus have developed resistance during the therapy of acyclovir because in herpes simplex virus, due to the mutation or because of the mutation, what happened? It become deficient for the thymidine kinase. So when there is no thymidine kinase, how the drug will convert into the how the acyclovir convert into its active metabolite. And for varicella zoster also is changes in specificity for viral surface enzyme, so it cannot detect it well. It is found that around 20% of an oral dose of acyclovir is absorbed. CSF concentration 50% of plasma concentration. Primarily excreted unchanged in urine by glomerular filtration and tubular secretion. And the plasma T half is around 2 to 3 hours. We'll see the uses of acyclovir. First use is against the herpes genital herpes simplex. By type, this is actually happening by the type 2 viruses. Can be treated by topical, oral, and parenteral acyclovir therapy. In primary disease, 5% ointment locally or 6 times a day for 10 days we can give. Severe and late cases require orally 1 gram per day in 5 divided doses or 400 mg TDS for 10 days. In addition, with the local therapy, means local therapy can continue along with the oral dose. In the case of recurrent diseases, topically ineffective, it has been found, and response to orally is also slow and incompetent. May be treated by parenterally 5 mg per kg IV, infused over 1 hour, repeated 8 hourly for 10 days. Prolong the therapy if reoccurrence also comes in. Continuous acyclovir prophylaxis is generally advocated in patients with more than 8 reoccurrence per year. If it is the case, decreases the transmission to, it has been observed during this case, decreases the transmission to sexual partner. Mucocutaneous herpes simplex infections is a type 1 virus disease, remains localized to lips and gums, does not usually require a specific treatment, but acyclovir skin cream may provide some relief. Spreading lesions may be treated with the 10-day oral acyclovir. Prophylactic oral therapy may prevent exposure related reoccurrences. The disease often gets disseminated in immunocompromised individuals and may be treated with oral or IV acyclovir 15 mg per kg per day for 7 days. But reoccurrence are not prevented. Third is the herpes simplex encephalitis. This is also a type 1 virus. Here acyclovir 10 to 20 mg per kg per 8 hours IV can be given for 10 days. Treatment is effective only if started early. Delay precludes salutary effect on mortality and neurological complications. Next is on using the herpes simplex type 1 keratitis. This is also a type 1 virus. Acyclovir is equally effective as hydroxyuridine in superficial dendritic corneal ulcer and may be better for deep stromal infections because of good corneal penetration. Though acyclovir eye ointment acts lower than hydroxyuridine eye drops, blindness can be prevented. The eye ointment should be applied 5 times daily for 3 days. Next is the herpes zoster. The Varicella zoster virus is less unsusceptible to acyclovir. As such, higher doses are needed and it should be used only if in immunodeficient individuals or in severe cases. Oral therapy with 800 mg 5 times can be given. Next is the chickenpox. In patients with immunodeficiency and in neonates only calls for specific therapy. Acyclovir 15 mg per kg per day IV for 7 days. So these are the uses of acyclovir. Now we'll see some potential ADRs as well as some contraindications of this drug. Consisting largely of headache, nausea. Actually, it is quite safe, as you have seen. Why it is quite safe? Because it's not acting much on the host enzyme. But even though there are certain adverse effects are there. So diarrhea and some less frequently observed are skin rash, fatigue, fever, hair loss, depression. Reversible renal dysfunctions, azotemia, and neurotoxicity like tremor, seizure, delirium. These are the dose limited toxicities of intravenous acyclovir. Adequate hydration and slow drug infusion can minimize the risk of renal toxicity. So, if there is a renal toxicity, adequate hydration and the slow drug infusion can be given. A potentially fatal disorder, thrombotic, thrombocytopenic, purpura hemolytic uremic syndrome, quite big name, thrombotic, thrombocytopenic, purpura hemolytic uremic syndrome, TTP-HUS, has been reported in immunocompromised individuals. Reversible neurological manifestations, tremors, lethargy, disorientation, hallucinations, convulsions, and coma have been ascribed to higher doses. Probenecid has been shown to inhibit the renal clearance of acyclovir. So there is a drug interaction with the acyclovir and probenecid. Cyclosporins and other nephrotoxic agents may increase the risk of renal toxicity of acyclovir. Here there are some drugs which you have to cover by yourself. These all are important drugs, especially femcyclovir, valacyclovir, gencyclovir, foscarnet. So please go through these drugs as well. These are the references given. Here I would like to just thanks to the frontliners who are fighting against the coronavirus. Just go through with this and wait for the part 2 of this. Thank you.